Thank you again. Es un placer de conocer a todos. Buenas tardes y bueno, it's a pleasure to be here and to tell you a little bit about the research that we do in my group. Okay, so today I'm going to talk about an application work that came about because a student was very much interested in nanopattern surfaces. And hopefully you'll see vignettes of some of the methods that we develop at the LFD with Enrico over the years. So my lab is really interested in cell migration, and we had several different questions about how cells are regulated in their extracellular environment, and they're controlled by their surrounding environment. We know that the extracellular matrix, or the ECM, the disruption of the basement membrane causes tissue stiffness, fibrillar collagen, like you saw before, not just in cancer, but in other diseases, such as fibrosis and kidney diseases. But we also know that the extracellular environment is also important for the cancer migration and invasion and their survival and proliferation. Let's see if I can go. So we know that the extracellular environment is highly controlled by their environment, the cells, but we were interested to understand if they can actually detect at the nanometer scale structures or topography that can actually change the behavior of the cells. So I'm going to give you a couple of stories here. The first one I'm going to talk about is the focal adhesion dynamics on nanotopography. These are surfaces that were made through nanolithography. And the second part of this vignette, or part of the talk, I'm going to show you changes of the metabolism in these tumor cells which is dependent on the extracellular environment. And finally, some of the work that we're continuing with is how contractility or forces, mechanoforces, are playing a role in this environment, and how the structure is really important for modulating cell behavior. So we know from the literature that micropattern surfaces or substrate topography can change cellular behavior and phenotype. So for example, this is work that was done in 2014 showing that if you culture cells in these micropattern surfaces, let's say micro lines, or let's say in this type of topography, cells will change their phenotype. They'll become more elongated along the lines, and without the lines, they will just spread out. We also know that in the microsurfaces, cells they actually move along the lines. So we know that the pattern itself, the topography, can influence not just the phenotype, but their actual directionality and behavior in the way that they migrate. So we were interested in nanotopography because we know that the ECM, the extracellular matrix, possesses topographical and adhesive features that range in the nanometer scale. So this is, you can find this in the membrane, the basement membrane of the epithelia and endothelia that has these kind of little, very, very small nanostructures. I don't know why I can't move okay, here we go. We also know that from the literature that the distance between the regions where the integrins can attach to play a critical role for this attachment of the focal adhesions, such that the cells can actually attach to the extracellular matrix and then migrate forward, push themselves forward for cellular invasion. So this is an experiment that was done showing that if you have, let's say, gold particles that are coated with the RJD, these are 3 amino acids that are critical for integrin binding, that only when the beads were close within the range of less than 70 nanometers will the integrins attach and form a focal adhesion at the sites. If the beads were placed too far apart, integrins will no longer form a cluster, and the actin cytoskeletal structure will not be tethered to the focal adhesions. So focal adhesions are really important. There are over 150 proteins that form these focal contacts in the cell. And from previous work, we've shown that focal adhesions have they're highly dynamic. So even though they look pretty static in images, they're binding monomer of proteins come on and bind to the focal adhesions. And as the focal adhesions detach, they fall apart in clusters. So our question was, can nanotopography modulate these focal adhesion turnover dynamics? So for our experiment, we took a library of nanopattern surfaces. Flat is just plain glass with no topography. And then we made these nanopattern surfaces. We made lines that had a array periodicity of 860 nanometers, meaning that the peak to peak between the lines was 860 nanometers. We also made posts, and the peak to peak distance between the posts ranged from 700 nanometers, 600 nanometers, and around 300 nanometers. And the smallest distance between the posts was this 200 nanometer post. And this post was actually a biomimetic that was carbon copied from a cicada wing. And this is considered to be an antimicrobial material. So bacteria can't actually grow on the surface. But what's interesting is that the closer the post became, the sharper the posts got, as you could see here in the P200. So as you already heard the lectures by others on number and brightness, I'm not going to talk about this. So I'm just going to show you that we use the number and brightness analysis to measure the dynamics of the focal contacts at the nano surface material. So we know that there are, let's say, proteins that are accumulating at the focal adhesions. Integrins are the very first proteins to establish a tether onto the extracellular matrix. Then proteins like talin, paxillin, vinculin are recruited at those focal sites. And we know that cells also have to modulate through that matrix, so they need to form invatopodia as well. But we were interested to see how the dynamics of these focal contacts change with these nanotopography materials. So the first thing to do is we measured the number and brightness of our cells cultured in just glass, and also in lines and in posts. And you see here all the images, representative images of the cells that were cultured in these different types of materials. And with the number and brightness analysis, we were able to quantify the population of molecules that were clustered at a given time point. So basically, we know from the previous work that if we see high clusters, that's where the focal adhesions are falling apart and disassembling, and that's giving us a turnover number. So from our data, we were able to calculate the ratio to monomers, so the population of higher order aggregates such as dimers and greater than 3 mers, and we saw a pattern or trend. So what we see is that the adhesion size on the nanopillars decrease with the nanostructure size, and the ratio of to monomers in terms of the dimer population decrease as the posts were getting smaller and smaller and smaller. The population, the percent population of dimers was getting smaller and smaller and smaller. And as you could see here, the population of greater than 3 mers also got smaller to the point where they were barely detectable in the P200. So what this was telling us is that as these focal contacts, focal adhesions are formed on these nanopattern surfaces, depending on the surface area, we believe less number of proteins can actually accumulate to form the focal contacts. And so when they disassemble in smaller clusters. So this was shown by the NMB analysis here. Now at P700, this trend dissipated, and we think that these posts were getting really large. So this was not part of the trend. And you could see here that the lines themselves look very similar to the flat in terms of dynamics. So we took these movies. This is a comparison between, let's say, tracking cells on these different types of surfaces. We see here the tracks of each individual cell. And what's interesting of the P200, and I'm showing you here, is that the cells actually have a high turn radius. So they're not traveling along anything. They're not just randomly traveling in any shape. They actually have this turn radius coefficient on this type of nanosurface on the cicada wing. And what was really interesting comparatively to the glass is that even on the nano pattern lines, the cells align themselves along the microgroups. So they changed their behavior and they did modulate their phenotype along this nano pattern surface, except for one cell. Every so often, we have a cell here that goes rogue and just goes off in its own way. And these cells are very interesting to study because it's almost like they're in a different class of cells. And that also is part of the heterogeneity of the cancer cells. So when we looked at the average speed of the cells on this type of material, we noticed that on the P200, the average speed of the cells increased by a huge factor. So we had increased population of the paxillin aggregates decrease with migration length. So we have smaller clusters that disassemble, and yet they travel the fastest in terms of speed. And there is a trend going down with the number of, let's say, percent of population of dimers and greater than 3 mers, with a change in speed here. So they're, you see they're anti-correlated. So we have that on the P600, the cells actually traveled the slowest compared to the P200. And that was a significant change relative to the flat cells. And so that was telling us that the smaller the focal adhesions, the faster the cells can travel, the more dynamic those focal adhesions are on that nano pattern surface. So the question that we were trying to raise with this whole study was, can nanotopography modulate focal adhesion turnover dynamics? And the answer was yes, and we're able to use number and brightness to answer this question. We noticed that the smallest nanopillars, which were P200, were the most motile. And the aggregation states decrease with decreasing nanopillar size. So as we got smaller and smaller, the aggregation population went down. And increasing aggregation disassembly results in greater speed. But we were interested to understand if the topography of the system also can modulate metabolism. So in this part of the work, we looked back into the literature and we saw that there might be a correlation between stiffness of the material and change of metabolism. So here's some work showing the total number of invatopodia. These are these tiny little holes that the cells puncture on gelatin. And you could see that the more rigid the material, in this case PAA, they increase the invadopodial let's say, dynamics. And we see that metabolism modulates cancer cell invasion. So we see that if you culture cells in different nutrients, for example, glucose versus galactose, for example, their degradation, right, those little holes that they puncture, increases depending on the nutrients. So it seemed to be that perhaps metabolism is influenced, the natural metabolism of the cell could be influencing cellular behavior in the extracellular matrix. So we wanted to put these two concepts together and understand could substrate stiffness regulate and shifts modes of metabolism? And so I'm not going to go all into the FLIM because you already had a whole day of FLIM lectures and the phasors on FLIM, but just important to note that the autofluorescence signal, as Suman mentioned, we're looking at just autofluorescence. We're not adding anything to the cell. This is the NADH in the free conformation and the bound conformation. They have 2 different lifetimes, and we can use these lifetimes to discern what metabolic states the cells are in. And of course, you know all about the phasors now. And of course, you can calculate the fractional contribution of free to bound NADH, giving you a metabolic trajectory for the cell. So one thing that we could do also, another method that we had, is we can correlate the intensity across the image and obtain, let's say, the standard deviation and the size, which gives us the size of the collagen structures, because in this case we're using collagen for the matrix, and we have a stiff collagen structure, 3 megs per ml and 1.2, which is very soft material. And we can calculate the modulus of this, so how dense the ECM is. And of course, you can guess already that 3 megs is more dense than 1.2 megs, but that was confirmed quantitatively. So what happens with the metabolism? And I'm just going to summarize this very quickly here. So we have tumor cells, and we actually put tumor cells on glass. Their metabolic state is highly glycolytic. They have a lot more free NADH. And as we get to softer and softer material, they switch their metabolism and they become more oxphos. In the MCF10A cells, these don't change. These are, as Suman mentioned, these are also tumor cells, but they're not aggressive. They have ER and PR positive receptors, and they don't care whether they're on soft or stiff materials, but our triple negative breast cancer cells do change their metabolism. So they're doing something very unique. And to show that this was indeed a change in metabolism, we had to use some inhibitors. We use a couple of inhibitors to shut down the oxidative phosphorylation pathway and to shut down glycolysis. So 2GG and DCA, and all you need to know about these inhibitors is what happens with the metabolism, and they behave as what you expect. With the MDA and B231s, these are the breast cancer cells, we see that adding rotenone antimyosin A, which is shutting down the OXPHOS pathway, makes them more glycolytic. So because they can't produce ATP through the oxidative phosphorylation, they'll switch to glycolysis. And that's perfectly confirmed here. And it's a very strong shift in these cells. So these cells respond even more than the the tumor cells that are not as aggressive. And I'm going to show you that in the next slide. And you could see that we can switch them back to more OXPHOS if we use the glycolytic inhibitors. Here's the data for the these are tumor cells, but not as aggressive. Their change is mild compared to the MB231s. So similarly, there is a change, a subtle change, and it's still significant, but it's not as much of a change. This confirmed that we were definitely seeing a change in metabolism of these tumor cells in this environment that we added. So the question was, are there other cells that are fine-tuning their metabolism? So we tried melanoma cells, and these melanoma cells on different substrates, glass, dense collagen, or not as dense collagen, they seem to have the same metabolism no matter what substrate that we cultured them on. So in summary, the metabolic inhibition shows glycolysis and OXPHOS are modulated in MB231 cells. We know that the material stiffness of the material does change the metabolism of the triple negative tumor cells. And now the question is, what happens in 3D space? So this is to show you the FLIM analysis, and this is some of the ongoing work because we're interested not just in 2D planes, but in 3-dimensional planes. We see that the metabolism seems to be a little bit more heterogeneous, specifically where the mitochondria are. And that led to a lot more studies on mitochondrial trafficking and dynamics. And so we believe that what's happening is that for the cell to push forward, to kind of drill holes through the matrix, it must be recruiting mitochondria to meet ATP demands. And I have a whole other lecture on mitochondria dynamics and trafficking. So if you're interested in that, I'm very happy to answer any questions about that. Unfortunately, I don't have that much time in this talk, but just to show you that in 3D collagen matrices, we see that, in fact, there is an alteration of oxidative phosphorylation glycolysis when cells are now cultured in three-dimensional space versus just on a 2D plane. So now that leads to, what is happening at the focal contacts? Can we put together the focal adhesion contacts and the changes of metabolism in this extracellular environment or in this nanotopography surface? And why is the metabolism changing? We believe that, and I think I need to stop. I don't have time to go on here, but I'm just happy to say there is one more last development from Enrico's group, and I'm going to highlight this because I think this is really important, and we don't talk about the Z phasors enough. This is a kind of, let's say, a cheap way to do super resolution in the Z-axis. So you've probably seen this famous picture here from the Waterman group, where they looked at the stacking of the proteins in the Z-axis. So we could do the same thing, but on a commercial microscope. So we used a Olympus FlowView 1000 to actually see the difference between integrin and paxillin, integrin, and any other focal adhesion, and to see how much they differ in the Z-axis. And this is really important because as cells are pulling, now this is the mechano-sensing part of it, we believe that as cells have to attach, they are, and they're going to migrate forward, or they're going to evade through the matrix, they need to put some sort of force on the cytoskeletal structure. And to see that, we've wanted to measure the distance between these focal adhesions. Okay, and I am going to stop, really, I'm going to stop. So we have integrin and paxillin on flat in those lines, and we do see a significant change in distance between the two focal adhesion proteins. If we add a ROCK inhibitor, they're no longer the change. All right, so we're using biosensors now, FRET biosensors, to see, let's say, the spring between these two proteins on the focal contacts. And I'm just going to leave you with the final note here, is that there is a lot going on at the molecular scale. And because cells don't just grow in 2D, they grow in three-dimensional space, it's important to characterize all of these kind of dynamics in 3D. So whether it's single cell-led dynamics or collective cell migration that occurs in cancer cells. And just want to